Hey guys, it's Medicosis Perfectionalis, where medicine makes perfect sense. Let's continue our biochemistry playlist. In previous videos, we talked about DNA, RNA, we talked about replication of the DNA, and we talked about transcription of DNA into RNA. Today, we'll talk about translation of what? Of the meaningless codons into meaningful proteins. Also today we'll talk about the post-translational modifications that are required to make your protein functional. Let's get started. Please watch the videos in this biochemistry playlist in order for maximum understanding and retention. The central dogma. I start with my DNA. Copy it, it's called replication. And then convert it into RNA, it's called transcription. In the beginning, we have heterogeneous nuclear RNA. If you want to make it into mRNA, you need post-transcriptional modification, which was the topic of the last video. The post-transcriptional modifications include splicing of the introns, splice them out, and add a cap at the 5' end, add a tail at the 3' end. Now your mRNA is mature and ready. Ready to do what? To leave the nucleus through the nuclear pores and to go to the cytoplasm, where it will get translated by the ribosome on the rough endoplasmic reticulum to give me amino acids, combine the amino acids together to make oligopeptides and polypeptides and then proteins. Modify your proteins to make them functional. And now your insulin is ready. DNA coding strand, that's the original sense strand. Replicated, you have the DNA template strand, which is a copy of the coding strand. Take that template and convert it into RNA. This is called transcription. Modify the H and RNA into mRNA, post-transcriptional modification. Take that mRNA and read it, i.e. translated, into meaningful proteins. The amino acids are made from the amino terminus to the carboxy terminus, from the N to the C. And whether we're talking about replication, transcription, or translation, we're always adding new nucleotide from the 5' to the 3' direction. How many amino acids do we have in the world? A lot. However, how many proteogenic amino acids that are coded by your genetic code? Only 20. We read the amino acids from left to right, from the N terminus to the C terminus, because this is the direction of their synthesis. Combine amino acids together, you have peptides. Combine peptides together, you have polypeptides and proteins. Dipeptides, 2 amino acids. Tri, 3. Oligo, few, and poly, many. So that's the sequence of events until you end up with proteins. How do I combine 2 amino acids together to make a dipeptide? Well, you will need to add a peptide bond between them. And as a result, we will sacrifice an OH from here and an H from here, and they will make water, which means it's a dehydration reaction because we removed water. It's also known as condensation reaction, and that's how you make a lovely peptide bond. It's also known as acyl substitution reaction. That's why we call it amino acyl tRNA synthetase. After we make proteins, they are not ready yet. We need some modification. And we need the proteins to acquire their structure. Primary structure is just the sequence. Secondary structure, now we have shapes. Tertiary structure is 3-dimensional. And quaternary structure is like my hemoglobin, 2 alpha chains and 2 heavy chains. We talked about all of this in detail in this biochemistry playlist. Types of RNA in this order. You start with rRNA because the most abundant. Then mRNA, which has the codons. After you have the codons, get the anti-codons on the tRNA. And in this order, who makes rRNA? RNA polymerase 1 in the nucleolus. Who makes mRNA? Number 2. Who makes the tRNA? Tree. Tree is for the tRNA, which has the anti-codon. Don't forget that the tRNA has the amino acid attached to its 3' end. Here is my lovely mRNA, which is the only type of RNA that contains genetic information that can be translated into meaningful proteins. And recall that in humans, it is monocistronic. 1 RNA molecule, 1 protein. Next, rRNA. Remember that the rough endoplasmic reticulum is smooth endoplasmic reticulum plus ribosome. What's the ribosome? Ribosomal RNA plus proteins. And that's why we call it ribosome because it has ribose sugar, not deoxyribosome because it does not have a deoxyribose sugar. What does the word "some" means? It means a body. From soma, site of birth of the ribosomal RNA, it is made in the nucleolus. Next is the tRNA, which we discussed in previous video. I'll just remind you of this reaction. Here is the tRNA. I'm ready to get activated and charged. Charged by what? By adding the amino acid to my 3' end. Who's gonna do this? Amino acyl tRNA synthetase. Any enzyme that's called synthetase will require ATP. tRNA has the anticodon, which pairs with the codon on the mRNA. Pause and review. Here is a mnemonic for you. RNA polymerase 1 is in the nucleolus because it makes the number 1 most abundant RNA, which is rRNA. So the mnemonic is you've got 1 rampant nucleolus. Here is transcription, which is conversion of the DNA template to hnRNA, which will become mRNA. After transcription, I have hnRNA. It's time to modify it. Splice the bad guys out, kick those introns to the trash. Keep the exons because the exons are expressed, but the introns, the bad guys, interfere with our job. Who caused the splicing? Spliceosome. Step number 2, add a cap at the 5' end. This cap is called 7-methylguanylate triphosphate. It is important because the ribosome will recognize the mRNA by the cap and will start reading from here, making methionine, the first amino acid. Step number 3 is the polyadenylyl tail, which also protects the mRNA. Rough endoplasmic reticulum versus smooth endoplasmic reticulum. The rough endoplasmic reticulum is the translator, i.e. protein synthesis. The smooth endoplasmic reticulum is the donut, or fat. Why is the rough endoplasmic reticulum rough? Because it is studded with granular ribosomes. The ribosome is made of what? rRNA and proteins. Rough versus smooth. In this video, we're focused on the rough endoplasmic reticulum. Protein synthesis or translation happens here on the ribosome, on the rough endoplasmic reticulum. And then a sequence of events will make us go up to the Golgi, package it, make a lovely vesicle which contains the protein, and then dish that protein, let's say insulin, out of the pancreatic cell. The mature mRNA will leave the nucleus through the nuclear pores, will go to the cytoplasm to find the ribosomes. Let's talk about translation again from the 5' to the 3' end. First, what's the ribosome? Here is the ribosome. It's made of what? rRNA plus proteins. Where is the rRNA made? It's made by genes in the nucleolus. Via RNA polymerase 1, you've got 1 rampant nucleolus. The ribosome has a large subunit and a small subunit. In prokaryotes, the large subunit is 50S. In eukaryotes, it is 60S. How about the small subunit? In prokaryotes, it's 30S. In eukaryotes, it's 40S. Unfortunately, students will try to add the large to the small subunit, and they will say, oh, 50 plus 30 equals 80. So why is this not 80? Because it's not math, it's not addition. This is just a number by ultracentrifugation, which depends on the size of the strand. So we say that prokaryotes in total have a 70S ribosome, not subunit, an entire ribosome. And eukaryotes, like you and me, we have the 80S ribosome. Each one of our ribosome has large subunit and small subunit. Let's talk about eukaryotes. Here's the large subunit called 60S. Here's the small subunit called 40S. Together, what's the name of the entire ribosome? It's called 80S. Let's dig deeper. The ribosome is made of what? rRNA and proteins. What kind of rRNA? Well, if you are in the large subunit, you have 5.8S, 28S, and 5S. If you are in the small subunit, you have the 18S. Who made the 5.8, the 28, and the 18? Answer, RNA polymerase 1. You have 1 rampant nucleolus. The other one that's going to help us to a minimal extent is RNA polymerase 3, the one that used to make tRNA. It will also give us the 5S rRNA, which belongs to the large subunit. Protein synthesis or translation is conversion of meaningless codons on the mRNA into meaningful proteins. You combine the amino acids together to make oligopeptides. Don't forget to join them via peptide bonds, a peptidyl transferase enzyme. Then you have oligopeptides, polypeptides, and then proteins. Are we ready? No. We need post-translational modification, and we need the protein to acquire its shape and structure. Hold your horses. Let's just talk about translation. It goes from 5 to 3 prime end. Translation happens in 3 stages: initiation, elongation, and termination. Initiation requires GTP because it needs energy and initiation factors. Elongation requires GTP for energy and elongation factors. Termination requires GTP and termination factors or release factors to release the peptide chain from the tRNA. Here is a side note. In prokaryotes, translation does not wait for the mRNA to be entirely made. Just make a piece of it, and before you know it, we're translating that piece. We're not waiting. You can say it's done prematurely. It's just a mnemonic. But in eukaryotes, you have to wait until the entire mRNA is done, until the mRNA leaves the nucleus through the nuclear pores, which are holes in the nuclear membrane, and then it goes to the cytoplasm, and then you can translate it. But in prokaryotes, recall, there was no nuclear membrane in the prokaryotes. So why wait? It's one of the reasons your bacteria replicate very quickly. Steps of translation: initiation, elongation, termination. Let's start by initiation, okay? Here is my lovely ribosome, P site and then A site. Just remember Pennsylvania, the P before the A. P is also prima, proto, all of these are words that means first. The first amino acid comes here, baby, the methionine. How about attachment of the second or the subsequent tRNA with the subsequent amino acid? It's going to go to the A site. And when you have 2 of them next to each other, here's the first amino acid, here's the second amino acid, link them together via peptide bond. So the first tRNA will go to the P site. Any subsequent tRNA will go to the A site. What's the first step in initiation? The small subunit comes before the large subunit, and the small subunit will attach here to the beginning of the mRNA, even before the cap, in an untranslatable region. In prokaryotes, this region is known as the Shine-Delgado sequence. In eukaryotes, this is the good old 5' cap, which is the 7-methylguanylate triphosphate cap at the 5' end. Because remember, the function of the cap is what? Is to be recognized by the ribosome as the first translatable site. And then you can translate the AUG into methionine. Who's the first amino acid? In eukaryotes, methionine. In prokaryotes like bacteria, it's N-formyl methionine. Mnemonic time. N because bacteria are nasty. Formyl because they are smelly. First amino acid will land here. The second tRNA loaded with the amino acid is going to land here. Then you make a peptide bond between them, hashtag peptidyl transferase, which is a beautiful enzyme that belongs to the large subunit of your ribosome. We're done with initiation. Let's elongate. We elongate in the 5' to 3' direction. The first tRNA loaded with the first amino acid methionine will land into the P site. Oh, by the way, and the last tRNA, which is hooked to the last amino acid, will terminate and gets released also at the P site. The beginning and the end happen at the P site. Mnemonic time. I call it the diapers mnemonic. From cradle to grave at the P, because you pee on yourself as a neonate and you pee on yourself as a senile geriatric person. It's always the P site. What does elongation need? GTP for energy, because making a peptide bond between this amino acid and this amino acid requires energy. So I'm going to get my energy from GTP. And I also need elongation factors, which are recruiters. They recruit the GTP and they recruit the correct amino acyl tRNA into its place. For example, here they will recruit the tRNA that's carrying methionine. I told you about the P site and the A site. There is a third site called E site. You know what's going to happen to this tRNA after methionine gets hooked up to the next one? This tRNA will leave the chat. It will exit the ribosome. How do you exit? You exit as an empty tRNA at the E site, which is more towards the 5' than the P site. So in order, they are E, P, A, not to be confused with the Environmental Protection Agency. Initiation, elongation, and termination. Termination requires GTP and termination factors or release factors. Why did you terminate the chain? Because I encountered a stop codon. Who encountered it? The A site did, which means we cannot put any amino acids here because that's a stop codon, which means the last meaningful tRNA was this one. The last amino acid was at the P site. From cradle to grave, it's the P site, baby. Okay, medicosis, how do you release this peptide chain that you made? Well, you need to add water, and water will cause hydrolysis of this bond. And boom, before you know it, this peptide chain is released. It's free to go. Thank you so much, release factors, for making this possible. Translation included initiation, elongation, and termination. After this, what happens? Post-translational modification of your polypeptide. And how can I do that? Well, post-translational modification is easy. You could add lipid, we call this prenylation. You can add a phosphate group, you will need a kinase, and we call this phosphorylation. You can add a carboxyl group, we call this thank you carboxylase, #carboxylation. Or you can add oligosaccharides, glycosylation reaction. Or you can add OH, hydroxyl group, hydroxylation. Post-transcriptional modification was splicing of the introns out and at a cap and at a tail. But the post-translational modification is not an and, it's an or. You can do phosphorylation or carboxylation or glycosylation, maybe more than one. But unlike post-transcriptional modification, it doesn't have to be all of them. Hey medicosis, why are we learning this? Have you heard of collagen, buddy? Oh, of course I know collagen. Type 1 is in my bone, type 2 is in my cartilage, type 3 is very flexible, so it's in my blood vessel, of course I know collagen. Without hydroxylation, i.e. post-translational modification of collagen, your collagen is absolutely useless. You know what's the name of the disease where I failed to hydroxylate my collagen? It's called scurvy. Without collagen, do you think I'll have robust bones? No. How about robust blood vessels? No. In fact, you're always bleeding from your gums or bleeding from any blood vessel. Why? No vitamin C, and vitamin C is important as a cofactor for hydroxylation, and hydroxylation is an important post-translational modification of collagen. You keep bleeding and bleeding and bleeding, you can even get abnormal hair because collagen is in your hair. To learn more about scurvy and vitamin C, watch my vitamin C video in my new biochemistry playlist. You know why you should care about post-translational modification? Because vitamin K is the hero of gamma carboxylation. What is carboxylation? Adding carboxyl group like what? COOH, which is COO. Or CO2, COO, that's a carboxylation. At the gamma carbon, which converts the glue to GLA. What's the function of carboxylation? Well, I'm trying to carboxylate so that I can bind calcium, and calcium is the hero of blood coagulation. So without vitamin K, there is no gamma carboxylation, which is a post-translational modification, which means calcium will not bind, which means blood coagulation will not happen. So what are the symptoms of vitamin K deficiency? Bleeding, because coagulation will not happen. See, medicine makes so much sense once you understand what the flip you're talking about. Learn more about vitamin K and vitamin K deficiency also in my Biochemistry new playlist. If you watched my previous videos, you remember the story of the headphones and how it gets entangled for no apparent reason every time you put it away in your backpack. That's called supercoiling, and it's a science called topology, which is a branch of mathematics. Your DNA is thin and linear. It is vulnerable to entangling, called positive supercoils. If your DNA is entangled, you are toast. You will never replicate, cell division will not happen, protein synthesis history, and you will die. Thankfully, you have someone to untangle it or preventing it from being entangled in the first place. Who's that hero? Topoisomerase. But what if we have a bacterial infection? How do we kick the bacteria in the nuts? Inhibit the bacterial topoisomerase. And this is how quinolones, one of the antibiotics classes, work. Medicine is beautiful. But wait, there is more. There is a difference between your ribosome, 80S, and the bacterial ribosome, 70S. Your large subunit as a human is 60S, and your small is 40. But the bacterial is 50 large and 30 small. Therefore, we can invent medications that target the bacterial ribosome without significantly affecting your own ribosome, because the bacterial ribosome is different from your ribosome. And this is how protein synthesis inhibitors, another class of antibiotics, work. And you can learn about these antimicrobials such as antibacterials, antivirals, antifungals, and antiparasitic medications by downloading my antibiotics course on my website medicosisperfectionalis.com. Next, we'll talk about the lac operon. So please subscribe, hit the bell, and click on the join button. You can support me here or here. Go to my website to download my courses. Be safe, stay happy, study hard. This is Medicosis Perfectionalis, where medicine makes perfect sense. No pun intended.